Hi learners, I am Professor S. Krupanidhi, heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research, a deemed university located in the rural vicinity, namely Vadlamudi, in the district of Guntur, Andhra Pradesh, India. And we are providing here a series of talks on immunology and immunoinformatics program, and in this, a few salient features of this title is immunology is a recent subject. It is being taught across all the universities world over. It is quite common. Whereas immunoinformatics is still more recent, it is quite uncommon. Particularly in India, a few institutions may be handling. But at our Vignan's University, we are providing this program of immunoinformatics for our students to get them trained in epitope, multiple epitope design and all those things. So here again, one more important feature I wish to let you know here is that we are making this program as semi-technical and semi-popular so that the students will follow this other than biotechnology students. So here I am showing you an animated video wherein a cell is getting infected with the virus. Once the cell gets infected with the virus, the cell doesn't keep quiet. It projects the components of the virus through a major histocompatibility complex. And that projection is being perceived by other cell called T helper cell. And this T helper cell produces interleukins. And these are the interleukins which are nothing but communicating molecules. As a result, one cell communicates to the other cell. So as a result, it communicates to the other cells and produces good many number of cytokine producing cells. And now if any other infected, it is not that we are getting infected with one cell, plenty of our cells are getting infected. Next time when any cell is getting infected, cytotoxic T cell, the name itself is called cytotoxic T cell. The cytotoxic T cell completely devours the infected cell, so that the infected cell along with the virus is getting totally destroyed in our system. So like this, the memories, cytotoxic T lymphocytes kill those cells. The next one is the B cell. B cell is the one which is playing a prominent role in our immunological memory. Whomsoever is speaking of the immune boosting means we have to improve our B cell population. So once the B cell is primed, then the B cell produces bailing out of immunoglobulins, an enormous number of the immunoglobulins. Those immunoglobulins bind to the virus or bind to the bacteria and form immune complexes. And those immune complexes are being taken by these cytotoxic T lymphocytes. And now the memory B cells are produced. This is what the immune potential. That immune potential is nothing but if I am having more number of memory B cells means I am now protected from the neighboring infections, neighboring pandemic pathogens. So with this video, we are now showing you how to improve our immune potential. So the objectives of our presentation is, in this first unit and first topic, to review the historical events in immunology. For any subject, there is a method of history is there. The history is the one which speaks the soundness of the subject. The second thing is to consolidate the sub-disciplines of immunology. And again, the third, the last objective that we are going to cover in this particular subtopic one is called to review, to overview the components of the immune system. So after having gone through around 15 to 20 slides in this first unit, first topic, so these are the following expected outcomes that we are anticipating from the learners. The depth of the discipline of immunology will be known in nutshell. This is only the first slide. Once we complete nearly 30-35 slides, the detailed depth of the discipline of immunology will be known to all the learners. So in the first slide itself, we will be having some information in nutshell. The second one is the contributions of Nobel laureates in immunology will be familiarized. The third one is the immunological memory to appreciate the vaccine will be known. Nowadays, everyone is speaking of the vaccine. Hitherto, we are not that much, what is it called, concentrating on vaccine. Now, every nook and corner in the world, people keep speaking on vaccine, the coronavirus vaccine, COVID-19 vaccine. So that's what the immunological memory is very much essential. What is vaccine? Vaccine is not something new thing that's coming. It is nothing but it's a virus only. So the virus they give, either attenuated virus they give, either weakened virus they give, a recombinant protein of the virus they give as vaccine. It's nothing but an antigen. Okay, so that antigen, once it is given, it primes our immune system. As a result, immunological memory is being maintained. All those details will be known to our students. And again, immunoinformatic tools to practice vaccine design will be acquainted. That is the last expected outcome out of this topic. System, in another 10 slides. See, our immune system is nothing but a defense system. As the country is having the defense system, our organs also are having a defense system. It's not that humans alone are having the immune system. Every system that is starting with the bacteria, protozoa, up to the vertebrates and humans, everyone is having the immune system. One important thing with regard to the immune system is those of the organisms wherein the immune potential is strong, their longevity is also strong. So that's what out of all the organisms humans are surviving for 100 years means our immune potential is stronger compared to other groups of organisms. So let us see now the tissues, lymph and lymph glands and lymph organs. Here we are seeing the thymus, and this thymus is the one most important in the immune system. As we said, pituitary gland is the master endocrine gland, thymus is the master immune organ, wherein the T cells are being produced. The T cells are said to be the master immune cells. And apart from that, this is ventral to the heart. It is located ventral to the heart. It is more pronounced during the infant stage. By the time we become the adult, this thymus gland is getting atrophied. Now the bone marrow is the one where the blood cells are being produced, spleen is the one where the cells are getting destroyed, and again the lymph nodes is the one where the immune cells are encountering the pathogen. Because our blood flow is not static, blood flow is continuously moving, where our immune cells are unable to attack the pathogens. Only in the lymph nodes the traffic is slow, blood flow traffic is slow, wherein our blood cells encounter the pathogen and develops the immune response. And then the cells involved. Another important aspect here is immune cells. It is not all cells are doing this immune function. Okay, so we have got the brain cells, liver cells, muscle cells. But exclusively the immune cells do this type of the immune function. They are immune competent cells, broadly classified, and immune auxiliary cells. The details we are handling in a different topic. Now again, in the immune system, we are having the molecules, immunoglobulins, antigens, cytokines, complements, and receptors. Here, these are the antigens. We keep speaking of the vaccine. Vaccine doesn't go without mentioning antigen. Clear? Now, let us see the historical events in immunology. If the history is sound, subject is pronounced. So that's what now let us see the historical events that is developing in immunology. Edward Jenner, the immunology made a beginning with Edward Jenner, who has done the cure for the smallpox, and the smallpox he has taken the mild pox called the cowpox to cure the virulent smallpox. So that's what. The next one is the Louis Pasteur. Louis Pasteur has developed some other type of vaccine, that is attenuated vaccine. For the first time in the history of immunology, the attenuation came into the picture from the Louis Pasteur. Next one is, this is the, what is it called, ampule for the smallpox vaccine. Now smallpox is not prevailing. It's totally eradicated from on the earth. Like that we have to eradicate coronavirus so that we should not suffer. And next one is the Ilya Michnikov. He is a Russian scientist. He has done the work on the phagocytosis. Phagocytosis, for the first time, a cell engulfing the bacterial cell is shown, and this is a part of innate immunity. The next one is Emil von Behring. He has done the serum therapy of diphtheria infections, and the serum therapy, what he has shown, those were the days. Now we are continuing as plasma therapy that's been spoken at length nowadays. Now, Jules Bordet has done a good amount of work on complements. The blood contains good many number of proteins, serum proteins, and the serum proteins, a few of them are dormant proteins, a few of them are silent proteins. Those dormant proteins are getting activated upon infection. That's what the complements. We are going to see those details in the coming lecture series. August Wieserman has done the syphilis, a complement fixation test, and Landstein have done the blood groups, ABO blood groups, and Rodney Porter and Gerald Edelman done on these antibodies. Nowadays, we are speaking much about the rapid blood test. Rapid blood test where in IgM, IgG we are evaluating. These are the called antibodies that's been done by Rodney Porter and Gerald Edelman. And McFarlane Burnett has done immunological tolerance. And now Murray and Thomas has done the transplantation. Nowadays, any organ is not functioning like kidney, eye, heart, they are getting transplanted. If at all there is matching of HLA matching is done. All that work was done by Murray and Thomas. And Rosalind Allo, a woman scientist, got Nobel Prize on radioimmunoassay. And nowadays, this is being modified as ELISA, enzyme liquid immunosorbent assay. At every nook and corner of the world, we can do the evaluation of this rapid test kit. The rapid test kit is nothing but based upon the ELISA technique. And Cesar Milstein has done the monoclonal antibodies. Nowadays, the subject of life science, biotechnology has come into limelight only because of the monoclonal antibodies, because they make a lot of economy for the country. And Susumu Tonugawa is a Japanese scientist. He has done this diversity of immunoglobulins, a good amount of theoretical computational biology oriented work. And Rolf Zinkernagel, he has done MSc restriction and antigen presentation. So this is one of the important aspects in cell-mediated immunity. Then George Snell, HLA diversity. HLA, that is human leukocyte antigen, is as many stars in the sky, so much of variation with regard to the HLA in human population. Myself and yourself are not having the same HLA. If we both share the same HLA because then your organs suit to me. So that's what nature has made in such a way that HLA diversity prevails in the human population. Then Bruce A. Butler, Jules Hoffman, and Rolf M. Steinman has done the work on toll-like receptors which work in this innate immunity, and James P. Ellison and Tatsuko Hunjo has done the blockade of immune checkpoint therapy to cure cancer last year, 2018-19. So with this background, milestones in immunology, will proceed further. Subdisciplines of immunology. So immunology, as we have gone through now, so there are several topics. Each topic is indicated in our syllabus at Vignans University. Now the subdisciplines are immunobiology, immunogenetics, and immunodiagnostics, and immunotherapy, and vaccinology, and immunoinformatics. This is what the new unit that we introduced for our students. Now, immunotechnology. This is also introduced for our MTech students. So now with this, now you see, just for the sake of understanding of a pathogen, in our blood we have got a particular type of the protozoan flagellate. This is called the protozoan flagellate. This is called trypanosoma. The trypanosoma is moving along with our RBCs, WBCs in our blood, and this trypanosoma till now we do not have the vaccine for this trypanosoma. Vaccines are being developed basically for virus, partly for bacteria, not for protozoa so far. Now people are suffering with the sleeping sickness with this trypanosoma. One of the this trypanosoma is so clever that its surface proteins keep changing. That antigenic drift is there in this trypanosoma. So overview of the immune system, let us say innate immunity. So I have been telling to you all organisms are having the immune system. But for us to understand, we need to classify this immune system as innate immunity and adaptive immunity. These are the two types by which we can classify it for the sake of our understanding. So innate immunity means it is pre-existing. There is no need to develop with experience. So it is highly non-specific. Any bacteria, any virus that we work, there are systems in our body that will attack this virus, bacteria. The adaptive immunity goes responsive. It is highly specific. Today, if I get infected with the coronavirus, my system develops antibodies against the coronavirus. And next time if I get infected with the dengue virus, our antibodies that are developed against the coronavirus may not work against the dengue virus. Again for the dengue virus, my system get used to that experience. That's what called adaptive immunity. So these are all the various pictures for you to get to know where innate immune potentials and this adaptive immune potential. Through the adaptive immunity, we are going to have antibodies. These are the antibodies that are specific to the pathogen, to the bug. So with regard to the innate immunity, any pathogen comes. So our complement system will attack, NK cells will attack. So that's what there is a built-in system in our organism, so that the organisms are having a continuous survival for nearly 80 to 90 years. Cells of the immune system. So hitherto I have been telling to you for the purpose of the classification, the innate immunity and adaptive immunity. Now let us see the cells of the immune system. There is a good many number of cells. Those cells are again subdivided into various cells. It goes to any length of the study with regard to the cells of the immune system. Let us see the phagocytic cells. Phagocytic cells are classified as leukocytes. Leukocytes are 4 to 5 types, of which the important one which I am showing is dendritic cells, macrophages, and monocytes. Okay, so these are the dendritic cells that are supposed to be the best antigen presenter. That means these are the cells which engulf the bacteria, or these are the cells which engulf the virus, and the virus is broken down and presents to what is it called, T helper cell. That's what the video which I was showing to you earlier. And then lymphocytes, and there is B lymphocytes. B lymphocytes are going to get differentiated as memory cell and the plasma cell. Okay, so this is the memory cell that will be kept aside for our future infection if at all we come across. And the plasma cells are the one which produce the immunoglobulins. That's what now, this is the term that we keep talking nowadays, plasma therapy. Okay, so the plasma cells produce huge many number of immunoglobulins. That means the person who has recovered from any of the infection will be having good many number of plasma cells. Apart from the plasma cells, the plasma cell secretions called immunoglobulins are also in bailing out positions, number of positions. And T lymphocytes, as I was mentioning to you earlier, T lymphocytes are the master immune cells. They are the one which regulate the total immune system. That's what whenever any person is infected with HIV, T helper cell is getting infected. Once the T helper cell is getting infected, the total immune system goes to what they call downtrend. That's what acquired immunodeficiency. Deficiency we speak here. So now the NK cell is the other cells, which is the lymphocyte. So now these are all the various cells of the immune system. The details of the cells of the immune system we'll see in the next unit. So now, just for the purpose of making you to understand how WBC cells, monocyte, will look into the bacteria. Now, as a man, I cannot see the bacteria. I require a microscope to see the bacteria. And again, I require a stain to see the bacteria. But that's not the case for the monocyte. Monocyte is one of the blood cells. So that sees the bacteria and projects its what is called pseudopodia. And again, so where is the eyes here? There's no question of eyes. Here it is all molecular patterns. Pathogen called the bacteria is having palms, pathogen associated molecular patterns. That is being detected by this monocyte because monocyte is having TLR, toll-like receptors, which I was showing earlier. So with this, monocyte looks into this bacteria. As a result, the monocyte are set to be scavengers of immune cells which are present in the tissues to remove the debris also. So there are two arms of adaptive immunity. One is humoral immunity, the other one is cell-mediated immunity. Humoral immunity contains antibodies. Cell-mediated immunity is being dominated by the T cells. T cells are nothing but cells which are produced in the thymus. So these are all the various humoral immunity examples where immunoglobulins are involved, and cell-mediated immunity is the one where cytotoxic T lymphocytes are involved. They kill the bacterial cells and the infected cells. So this is the central dogma of immunology, wherein, so if any antigen is nothing but a foreign body, a foreign protein, a pathogen. If that comes into our system knows, we may not be knowing. That's what the doctors say, keep yourself aloof, keep maintaining good health practices. But our system knows what is antigen. So as a result, either macrophage or dendritic cell engulfs the antigen and promotes T helper cells. And T helper cell in turn through interleukins promotes B cell. B cell produces antibodies. Okay, and these are the antibodies that neutralize the antigens. Okay, now the next question is how long the B cell produces antibodies is a big question. That it is a feedback information. Once the neutralization completes, the feedback it generates, and as a result, the T helper cell prompting the B cell stops. So this is what happening in the immune system, immunological memory. So immunological memory is more rapid. So this is what called immune potential. This is what people keep speaking about, boosting your immune potential. If you boost your immune potential means your immune memory is rapid, your immune memory is intense, your immune memory is prolonged, and your immune memory is specific. That's what as I've been telling to you, that if I get infected now with coronavirus, my immune memory is specific to the coronavirus, not to the dengue virus, not to the Japanese encephalitis virus. Okay. Now the clonal selection theory. See now the memory. So see what happened, we are not having one B cell, we are not having one T cell, good many number of B cells are there. Which B cell encounters the antigen? Which B cell encounters antigen is a question. If that B cell encounters antigen, that B cell claves, divides with the clone, we will be having clone of cells. And this clone of cells produces immunoglobulins. So these are the immunoglobulins which neutralize the antibodies. So that's what the clonal selection theory developed by a nice German, a Danish scientist who was given the Nobel Prize. So this is again the CD4 T cells. Whenever a person is getting infected with this HIV virus, that is, let us say he is infected on the 3rd week, his CD4 T cells are 1200 count. Okay, so now as the infection keep progressing, by the end of the year, the T cells are coming down to 600. And due to unknown clinical latency, this infection prevails up to the 6 to 7, 8 years. After this 8 years, the T cell population keeps going down. It is dwindling to 100. Once the T cell population goes down, then he will face opportunistic diseases. That is, some other infections he will get as it is, because T cell is said to be the masked immune cell. If there is no masked immune cell prevailing, he is exposed to the other opportunistic infections. So as a result, the death occurs after the infection, nearly after 10 years of the infection. So that's what here again the T4 cells are involved. So the clinical mediators are in this immune system. That's not only the cells involved, there's not only the organs involved, there are chemicals also called the cytokines, which constitute the chemical messengers. Chemical messengers means one cell communicates to the other cell. As I am now communicating to you to understand the immune system, the dendritic cell is said to be the best antigen presenter cell, presents to T helper cell. So that's what the next one is, the complement system. This is also a sort of innate immune aspect of the immune system. Okay, so apart from that, this immunoinformatic tools that is being taught at our Vignans University, these are all the various tools we are now giving training to our students. This iterative thread assembly refinement, and then the Vaccgen, and again the Grammix, and again the B cell epitope prediction, and again immune epitope database and analysis resources. One of the awesome tools is the SNAP gene, wherein the gene of our choice we can insert into the plasmid, and again we can work on this plasmid. The other important software tool is computerized immunological simulation tool. So with this tool, we'll work out all simulations of the immune system once the organism is getting primed with an antigen. So like this, we'll work with this type of informatic tools. So this is one of the ill health practices that most of our indisciplined people are doing. So as a result, we are spreading the infection to the neighbors. This we have to avoid. So now, with this few slides, now I introduced you this immunology and immunoinformatics course that we are handling to our students, wherein the summary of this particular 15 to 20 slides is cellular and biochemical components of the immune system are known. Historical events showing the milestones in immunology are illustrated, and subdisciplines of immunology and an overview of the immune system are made familiar. Immunological memory and immunoinformatic tools are made aware. And a few study questions before we come to the next topic. Please go through the slides and come to an understanding. Name the tissues involved in the immune system. Name the cells participating in immunity. Enlist at least 10 milestones in immunology. How is immunological memory developed? What are the ill practices in spreading the diseases? enumerate the immunoinformatic tools. So with this, I wish that I have given you sufficient information related to the topic 1 of Unit 1 of Immunology and Immunoinformatics program. Please go through this and again we'll meet for the second topic. Thank you one and all.